All right, so last week we talked about transcription, which is the process of taking that DNA and transcribing the message from that DNA onto the mRNA, which is smaller and can actually leave the nucleus. All right, so just a little review. If our DNA strand says TAC, our first codon for our mRNA strand would be AUG. Just a reminder that when doing mRNA, we do not have any thymines, we only have the uracil. All right, so then this one, GCT, would then be CGA. CAC would become GUG. AAA would be UUU. CGC would be GCG. And then ATC would become UAG. Okay? All right, so today we are actually, and this is on page 87 in your notebook if you want to follow along, we're going to be ending up finishing up these two questions down at the bottom. But today we're going to be talking about this next step. So taking this mRNA, translating it into the transfer RNA or tRNA, and then coding for the amino acids that come from that. So before we can get started, we've got to kind of look at the overall process of protein synthesis. Okay, so when we're looking at protein synthesis, and this is the easiest way to draw it, I'm going to turn my paper to the side, zoom out a tad so we can see this. All right, so with our protein synthesis, we always start by looking at the overall process, which begins in the nucleus. All right, so within our nucleus, and I'm going to label that. Within our nucleus, we have our DNA, all right, so our double helix. All right, but remember that DNA is too big, it cannot leave the nucleus, so we've got to have the mRNA to come in and make a message from the DNA. All right, so we take this mRNA, we come in, we've got our open mRNA, and this is where we start to see that transcription process. So I'm gonna label our mRNA here, and this is the process of transcription. So transcription is happening inside the nucleus. All right, so once that transcription has occurred, then that mRNA is going to leave the nucleus. So let's draw that mRNA leaving the nucleus. All right, and that mRNA is going to go to our next big structure, which is a ribosome. All right, and the ribosome is where protein synthesis is truly occurring. So here's our ribosome, let's label that. So ribosomes are responsible for the creation of the proteins themselves. All right, so we've got this ribosome, we've got our mRNA coming straight through it. We've got our code from the DNA written on this mRNA. And what's going to happen at this point is that our tRNA is going to come in and it's going to start to drop the corresponding codons. So for example, let's look at our mRNA right here. Let's get ourselves a codon. Remember that a codon is 3. So our codon right here is our 3 bases. Let's say that this one is AAA. All right, so just to make this simple, here's our little tRNA, and they look kind of funky. Here's my best impression of them. All right, and they have what is referred to as an anti-codon. So anti just meaning the opposite. So if we have AAA here, then the opposite of that must be UUU. So this anti-codon on this tRNA is going to fly in, and it's going to drop that UUU and attach to it. All right, but what's crazy is that this tRNA, let's label this as our tRNA, our transfer RNA, has this little thing attached to it. And this little ball that's attached to it is an amino acid. And amino acids, if you remember, are the building blocks of proteins. All right, so the cool thing about this is once that UUU has come down, it's attached to it, then it drops that amino acid. All right, so for example, UUU, it may be the amino acid, this, okay? So then maybe the next codon that is coming through is going to be, I don't know, GCG. All right, so then that tRNA that comes in and attaches to that one would have the anticodon CGC. And it would carry a different amino acid which would then be dropped down and start attaching to this. So as this mRNA is running through the ribosome, we're going to start to see that it's building this chain of amino acids. And this chain of amino acids basically is a protein. All right, but we refer to this as a poly. So poly means many. Peptide, and peptide is just the bonds in between the amino acids. Polypeptide chain, also known as a protein. And that is the overall process of protein synthesis. All right, so now we've got to go in and we've got to figure out how we're going to calculate out which amino acids are being dropped down. So there's a really cool thing called a codon chart. And here's your codon chart. So going back to page 87, here's your codon chart. And I really like doing this. I'm going to use 2 markers in order to show you how to work this codon chart. Okay, so on page 87, I like this codon chart. It's really big, really easy to see. There are a couple of them in your handbook, but this one's the one that I'm going to use today. Okay, so we are going to look at our mRNA only. So this first mRNA says AUG. Notice that none of the letters on the outside of our codon chart are T's, meaning that you should never, ever, ever use the DNA strand. You should only be looking at the mRNA strand. So I'm going to highlight that in pink. This is the one that you should be looking at. Okay, so the very first one we said was AUG. So let's look at that one. A, U, and then G. All right, so let's one more time. Let me zoom in. I'm going to show you how to do this. So the first letter, the second letter, and the third letter are how you're going to use this. So the first letter was A. So we're going to take our first letter, we're going to drag all the way down till we find the A. Okay. Our second letter was U. So we're going to find the U on the top. Okay. So now we've got a point here, a point here. All right. And then our third letter was G. So in our third letter category, we're going to go all the way down to where all of these intercept with each other. So to make this easy, I'm going to use some colors. All right, so our first one is A, so it could be anything within this row. Our second letter was U, so it could be any amino acid within this row. Notice that now they intercept. So the only box you should be looking at is this box right here, because that's where letter 1 and letter 2 intercept. Letter 3 is G, so we're going to look at the G, and the only row that it can come out of is this row. Therefore, the only square that all 3 intercept in is this start codon. All right, and we like to, in science, make it a little bit easier, and we're just going to abbreviate that. So we like to take the first 3 letters of our amino acids because they have some crazy names, and I do not expect you to have to know these names, and we're just going to write that down. So the first 3 letters, well, yeah, I'm going to use the first 3. First 3 letters are M-E-T. So that would be your first, I'm sorry. All right, my last video got cut off there, so I'm gonna pick up right here and I'm gonna piece these back together. All right, so our first one, our MET, would be where we write in our amino acids. And notice that I did leave the tRNAs blank. It is up to you whether you want to do those. It is a great practice to figure out what the anticodon is. So basically for this line here, for the tRNA, you would just be telling me what that anticodon was that matched up with the mRNA. However, for the purposes of the amino acid chart, notice that it says mRNA codon chart. You're only using the mRNA strand in order to do the amino acids. So let's do the next one real quick. All right, so the next one is CGA. All right, so let's zoom out. Let's look at our chart a little bit. All right, so our first letter is C right here. Our second letter is G right here. And our third letter is A, which is right here. So ARG is our second amino acid. All right, so now let's kind of keep going through these and get a little bit of practice and see if we can do this.